Hey, this is Nick at NASS 2023 in LA. In October, I'm here with Jordan Katz.
Orthobond is our company.
Okay.
We are a material science company. Our product is an antimicrobial surface, which we call OstaGUARD. We've been developing the surface for over a decade now, and we are at the precipice of commercialization, so excited to talk to you about it.
Yeah, I'm interested to learn a little bit. And so, if I understand, it's a mechanical lysing process to be antimicrobial, but it's like a mechanical process of killing microbes.
Yes, so we look at a surface as chemists and chemical engineers, and we take the oxide layer and actually perform covalent permanent chemistry on the oxide layer to apply about a millionth of an inch of organic chemistry to try to change the behavior of that metal alloy as a biomaterial. Our sole focus has been on turning these materials into antimicrobial surfaces, and we do this by putting a high density of positive charge right
Oh,
at the surface. Bacteria have a net negative charge, opposites will attract, and once the bacteria are in a close proximity with our surface, multiple mechanical mechanisms of action will start, where the bacteria cell membrane is essentially torn apart. This gives us rapid, broad spectrum antimicrobial activity, helps us avoid bacterial resistance, which is a major concern when it comes to antimicrobial medical devices.
what was the process to get to where you're at right now?
So we have spent about a decade on this, and I like to bucket it into thirds. The first third of the decade was really, how are we going to kill bacteria on a surface without releasing metal ions or any antimicrobials into the human body? Really, how do we develop a technology that maximizes reward and minimizes risks so that we've got the possibility of making it through the FDA? So
Mm-hmm.
3 years just to figure out how to do that. We tried thousands of technologies or combinations of covalent linkers and antimicrobials. We found a class of molecules which present a high density of positive charge, which did the job.
Okay.
Second 3 years, pardon me, we then focused on generating all of the safety and efficacy data in order to be able to put a submission to the agency together. And now where we've been on this last third of the journey has been presenting the data, discussing it with the FDA, making them comfortable with what the surface does,
Mm-hmm.
arriving at claim language that we can support with data, and then carrying through a review process.
So, where are you in the process with the FDA?
So we are in the middle of a review on two technologies.
Okay.
One is a pedicle screw system, where
Oh,
we
okay.
have a partner who is here, and we hope to announce them shortly after we get our approval through the agency. That technology is about 3 months from decision, and we have another partner in orthopedic oncology,
Okay.
and they are on a similar time frame. So we're getting to the end of our review. That's why we're here at NASS, sort of to announce ourselves to the world,
Yeah.
and make people aware of what we're doing.
Okay, that sounds great. So, tell me a little bit about the company. You mentioned that the work on the technology has been in process for just over a decade, I think is what you said. Um, Tell me about the company a little bit. Like, what's the origins, that sort of thing.
So our name, Orthobond, we were actually founded as a company with the intention of making a bone-friendly surface. And it really all came out of conversation between a physiatrist from HSS and a chemist at Princeton. They were talking about how most of the metal biomaterials don't perform that well in terms of bone integration. And the chemist said, I think I can modify the behavior of the metal alloy. So the first early part of our company's existence, there was a focus on trying to covalently modify that surface to improve its osteoconductivity.
Interesting.
What we found in those early animal studies was that we could get a transient improvement, But over time, the controls and the treated groups, really, there wasn't much difference.
Okay.
But the one thing that we observed was that the linker was absolutely stable in the human body. And so we decided to make a pivot into the antimicrobial space because we knew that if we could actually create a surface that doesn't deliver the antimicrobial, we could avoid being a drug delivery device.
Right.
And have really a unique technology minimizing risk to the patient. So we made that pivot, spent those first 3 years figuring out what would work on that surface. But that's really how we were conceived, and then we moved into the antimicrobial space.
Okay, interesting. And the goal at this point, there's a threshold of activity or of active response that you get from the coating. I'm assuming that once it's implanted, then it's over time going to lose its effectiveness locally, right?
So by doing covalent chemistry, we are putting this millionth of an inch of organic on the material. In the human body, there is no such thing as a permanent organic material. But what we've been able to demonstrate with our safety testing is that as the device integrates in the body, what comes off of the surface is degradation products, and that those degradation products are safe. The antimicrobial activity is really designed to occur from package to patient. There is a lot of data in the scientific and clinical literature that shows that most bacteria are contaminating a device from the time the surgeon opens the package to the time they close the wound.
So
And
like aerosol?
aerosol contamination from the surgeon's gloves, some from the patient's skin. This is where most of the bacteria gets on the device, and it's also when the implant is most vulnerable. A lot of papers have shown that if only a few hundred bacteria get on that implant, then a device infection can occur. But it takes millions of bacteria on a wound without an implant to get that local infection. And so our concept is if we can reduce or eliminate the contamination during surgery, the doctor can close with a clean wound and improve the chances for healing to occur on that wound site.
Yeah, that's interesting. So you're here at NASS. So obviously the goal here is to connect with companies and people working in spine. Are there other applications or are you focused on spinal implants at this moment, or are there other applications you're interested in working in?
We are a material science company, and so we chose spine because there is a series of papers that have come out the last few years showing that contamination of pedicle screws intraoperatively is a big problem. But our company, we've got a partner in trauma, in orthopedic oncology, in neurostimulation. We're talking to people in the cochlear implant space, as well as the cosmetic and plastic surgery space. Really, bacteria on all of these implants across medicine present a huge challenge, a huge risk to the patient, a huge cost to the healthcare industry, and we see our technology as a platform that can really make a difference in all these different spaces.
So this is the first trade show that you guys are doing this with? Are there other trade shows that you're targeting, I guess?
We will go to the AAOS.
Okay.
Now that we've branched into some different areas, we're looking into additional trade shows. So cardiovascular, dental. So a lot of opportunities, new places for us to go with the technology, and really the thing we've heard in all those spaces is the need from the end users, the doctors, for strategies to help them address bacterial contamination.
Okay, that's great. Well, appreciate the time, and we'll be seeing more of you hopefully.
Yeah, thank you.